Hi, this is Miss Litton, this 7th period with my wonderful APBio students. Say hi.
Hi.
This is the beginning of the end. We are going to review through all the big ideas. How many are there? 4. And the first one's on what? That's right. It drives the diversity and unity of life. So what we want to start doing in these big idea reviews is not thinking of things as units, because your AP exam certainly won't think of things as units. They're going to pull from all over the place. So we're going to pull from multiple chapters. At this point, sometimes I'll bring up something maybe we haven't studied yet, but we will study, but it's under the umbrella of this particular topic. Okay, so I want you, before I even start, pull from your brain. It has to be different. Ping pong twice. Something about evolution, key factors, key somethings about evolution that you can share with your bio buddy. Start to unleash the evolution memories. As soon as he's done, you can take it. I had it set up for you, but he walked in first. I called for you, so put your name on that. And when he's done, you can have it.
All right, thank
Jake, are you playing games in English? Talk to the hand. All right, now.
That's on tape.
I'm recording.
Oh my God.
Thank you, baby. All right. It's a family here. We know all. All right. Oh my gosh! Look at all those beetles. Okay. What do we remember about those beetles? They're all different. Why might they be different?
Adaptations.
Yeah, different environments, different adaptations. Wait, I don't remember what that link is. I'm just curious. Oh, that's where I got it. Okay, sweet. All right. Sorry about that. I was like, what did I put there? Now, do you remember what this was about? Microevolution? What is microevolution? Within that population, within that species, not necessarily population, within that species. Macroevolution, girls, is when you change to entirely what? New species. Good. So underneath the big idea evolution, there are enduring understandings. You have 55 enduring understandings that you want to be familiar with. How could you find 55 videos on the 55 enduring understandings? Bozeman. He has them, Mr. Anderson, has them down 1 through 55. Okay? And you can review all 55 of these enduring understandings. So the first one is that change in the genetic makeup of a population over time is evolution. So when you hear things like change in genetic makeup is evolution, you should be thinking about population genetics, right? And a couple of equations should come to mind. A couple of equations should come to mind. What are they? P plus equals 1 and P squared. What are the words to describe that? HARDY-WEINBERG. Good job. Okay, must have got that from your game. All right, now, so when we think about a mechanism for evolution, we're thinking about things like natural selection. And Darwin came up with that particular mechanism, yes? And then when you look at what the premises of that is that you have more, the struggle for existence, you have more offspring than can survive. The struggle is real. Okay, so not all of them are going to make it. When you look at all of the offspring, there are what in them? Variations. And some of those variations are in their DNA. Good. In their genes. If it is a variation due to their particular environment at that particular time, or because something happened to them, you're welcome, in utero, and it's not actually in their DNA, that's not something that natural selection can work on. It can only work on something that's actually in the DNA, right? Okay, so if it's in their DNA, then that's something that can be selected for. But it has to play itself out in its what? Phenotype. Phenotype, good job. And then that particular organism would survive over time. So your first step is that you have to have variations, amongst the offspring. have to be some differences. We talked about population genetics, remember when we did this? And we talked about the equations, right? So let's break down those two equations. What were they again?
P plus Q equals 1.
And?
P squared plus 2PQ plus Q squared.
Were these those equations? Yes, they were. So when you're looking at this second equation, these are, you're looking at pheno and genotypes, right? Do you agree with that? Because this equation right here, both of these would show what? Yeah, they would be the dominant trait. Okay, the p squareds and the 2pqs. But the q squareds, you would know the genotype and the phenotype and the genotype, right? Because there's only one way to be that. So that's why if they give you, and they say, oh, 85% of the population has the dominant trait, you're going to dismiss that and you're going to focus on the 15%, the 0.15, because that's the genotype you would be able to figure out from that. You can't figure it out from the dominant, because those could be p squareds and the 2pqs. Once you have, you know the q squareds, right? If you said q squared equals 15%, how would we write 15%? 0.15, right? Then what would I have to do? Take the square root to get what? Q. So if I take the square root of this, I have Q, then I can solve for P. Once I have P, I can square it to get this and 2PQ to get this, and I just have to multiply it by that particular population. Yes?
This is going to be as good as question. I don't have to make like a plus or minus for the root 15. It's just always, I can't be negative.
Yeah, right. Right. Yes?
It is.
Oh, you're just waiting? Okay. So, the Hardy-Weinberg law is set up basically for you to break it, right? Because it's saying that if evolution, when evolution occurs, it's because allele frequencies change. And if you can freeze the allele frequencies, then what? No evolution. And it's difficult to keep those allele frequencies where they are because different things can affect it, right? How many different things? 5 different things can affect it. And so anything that changes it will cause evolution. So we looked at, I don't know if it's going to transfer over because this is my old review and it's, but we'll try. Let's try. Wait, let me remove these students.
The same code.
Let's be famous athletes as opposed to the infamous athletes. You can be an infamous athlete too. You can be your best friend who runs on track. You can be yourself. Somebody's Sriram.
The most famous athlete I know is Dave. That's all I'm gonna say.
What? No, I feel like that's wrong.
Is this right? Ours or yours?
All right, I don't even know if this question's going to transfer. We'll see. I can't. Is it working for you? Can you read the question?
Yeah.
4%.
Don't do it out loud. Calculators are in the back. I'm going to pause this while you work through it.
You have your phone that has.
Okay, I got all of yours done. Are you done? Yeah, 0.2. 0.2 obviously works. It's just however I put that answer key translated over. Okay, so 4% of the members of a population of pea plants are short. That's a recessive characteristic. What is the frequency of the recessive allele? How do you write 4%? 0.04. And when you take the square root of that, what will it be? 0.2 or 0.2. Okay? Yes?
Wait, because I just had that realization. So,
Guys.
I know it's a while ago, but so the P plus Q is the amount of those, like, the recessive and dominant,
Okay.
and then the...
Okay, so hey, can you hang on to that for a second?
Yeah.
Okay, so if I had a plant that was tall, okay, and another plant that was tall, and a plant that was short, and a plant that was short, and here's a tall plant. Okay? So, to be short, what are you? two what?
Little t.
Two little t's, okay? Let's say this tall is like this, and this tall is like this, and this tall is like this. You with me on that? Okay, so the gene pool is all of these t's, right? So how many t's do we have total here? 10 of them, right? 10 t's. How many big t's do we have?
5.
Okay, so 5 out of 10 are big T, so that means we don't even have to count them. What is it? 5 out of 10 are little t. You with me on that? What letter do we pick for big T? P. And this is what? Q. So this would be when we do P plus Q equals 1, right? Then that would be 0.5 plus 0.5 equals 1. So 50%, that's looking in our gene pool. If everybody took their genes and threw them in the gene pool, and we're diploids, so we have 2 copies. Everybody takes their genes, throws them in the gene pool, and we count up all the big T's and count up all the little t's. What percentage is it? That's what the P plus Q equals 1 is. P squared would say we are predicting then that the big T would be 25%, right? We could predict that. It doesn't mean that's how it played out, but we would predict that. So 0.5 times 0.5 is, we would predict 25% are big T, right? Okay, and 25% are little t. That's what you would play out from that. It doesn't mean that it's reality. In this case, I gave it to you exactly what it was. Okay, got it? All right. Now, so we said mutations is another thing that can cause a change. Okay, and can we control mutations? Nope. Okay, we said gene flow. What is gene flow? New blood coming in or somebody going out. And remember, we talked about ways you could lose genes. We talked about genetic drift. And we talked about the bottleneck effect. A bottleneck effect is whenever there's a what? Natural disaster, a crisis, okay, that we're going through. What was the other kind?
Founders.
Founders effect. Good. Oh, here I have another one. Here. So founders effect is if you look to see, if you look at our classroom right now, do you think we're a representative of the general population of Oak Park High School?
Well, maybe
So I don't know that we're, it'd be interesting to see if we are, but we don't, if we went and formed an island and we were the produced a whole from our island, okay, there's not very many girls. Okay, if we produced everything from our island, we got more. Then what would that be representative? Or maybe some of you are harboring some trait that's negative, and because we only have ourselves to interbreed with, and we would have more of that particular trait showing up on our island. That would be an example of founder's effect. Both of those genetic drift. All right, so natural selection can cause a change where nature selects, and we looked at artificial breeding would be something that's already evidenced when people select for certain traits, whether it be plants or animals, or some dogs. We talked about different kinds of selection. Directional selection is what? Yeah, one extreme or the other, right? Could directional selection lead to speciation? Yeah, it could lead to a totally new species, okay? And then we talked about stabilizing. Could this lead to a new species? What do you think? I think no. Because you're selecting against either what? Extreme. And you're saying this is best. It's like the size of babies that are born. All babies are around, 7 to 8 pounds. Are there ever 13-pound babies and 4-pound babies? Sure. But the healthiest babies are in the 7-8 pound range. The number of eggs that a certain bird lays, maybe it always lays 3. Maybe another one always lays 5. That's stabilizing. It's not looking for any differences. It's trying to keep it same. I saw a hand, yes.
At what point is it changing the species and making a new species?
Ah, that was the big question, right? Remember we debated that later. Remember we talked about the different kinds of isolation? started with habitat isolation, we talked about temporal isolation, we talked about behavioral isolation, and at what point, and that's the point when they don't breed anymore, that they're genetically isolated. And we talked about maybe they breed, but the zygote doesn't develop normally, or maybe the F1 generation lives, but the F2 generation doesn't. So that's a hard one, because a lot of times you'll have species right next to each other, and maybe they'll interbreed with each other, but 2 or 3 species down they won't. That is a hard line. Most biologists don't care though. It's, you know who cares? Is if somebody who says there is no evolution. You know, they're going to say species are unique, but it's like you're saying, well, I don't really care. This one might breed with this one. Maybe it forms a new species. Maybe it doesn't. It's not as big of a deal. You know what I mean? It's like generally these species, they breed together.
And theoretically, if over enough time, if like certain humans mate with other kinds of humans, would it be like a diversion, like human beings, like 2 species, kind of like?
Probably not 2 species. We would all start to look the same.
No, what if only one group of people only like mated with that group of people? Is it possible for like a group like humans to mate to diverge in 2 species?
There would have to be some advantage that made them better, where they couldn't go back and mate with another one. There's no humans that can't mate with each other. There's nothing reproductively isolating, so they can't. You could say there's social norms, which I would put under behavioral isolation. But if you could still mate, if the room was dark and you couldn't tell, you could still mate with them. All the parts still work together. You see what I'm saying? Okay. All right. Disruptive selection. This would be like, it's favored if you're a dark-shelled snail or a light-shelled snail, but maybe if you're an intermediate colored snail, you show up more. You can't hide as well. Could this lead to speciation? Yes, this could. Okay, this disruptive speciation, or what is it called when you go to one direction? Directional. Those could lead to speciation, whereas stabilizing usually wouldn't. Okay, so you can promote variations. And why do we even care about variations? Variations?
Evolution.
Yeah, we need variations for evolution to occur, right? So mutations, gene flow, natural selection, things like disruptive or directional, these types of things can lead to variations, which can lead to one particular phenotype being better than another. So then we talked about the heterozygote. So in order to be selected, it needs to be expressed. So even if it's a bad trait, if it's a dominant trait that's lethal, it's probably going to get removed fairly rapidly. But if it's a recessive trait, it's going to be what? Hidden. Can we ever get rid of it? Are your knees getting sore? I'm starting to stress out about your knees. Do you want to share? Okay. So if it's a recessive trait, can we ever get rid of it? Can we? Yes or no? No. Why? Carried in the heterozygote. Yeah, you'll probably never completely eliminate it. It can always be sheltered. All right. Evidence of evolution. Could you give evidence for evolution?
Yes.
Okay. So what's this one? Fossils. What did we learn about fossils?
Must be
10,000 years old, so close. Okay. But what did we observe in the fossil record?
Oh, there's species that don't exist today, or species that are in between 2 different environments.
Okay, perfect. You said 2 of them. Yeah. What? Transitional. We're getting it there. Okay, so there are species that don't exist now that existed in the past, and it looks like there are transitional forms. Not always, right? But oftentimes they're found. We talked about the process of fossilization is difficult, right? So to be a fossil, you have to be some hard structure that can be preserved in the right environment, not eaten, not destroyed, right? So just the process of fossilization is difficult. And then once that fossil is made, it has to be found by somebody and characterized and said, this is this fossil, right? So that process is difficult. Okay, so then what's this one right here represent? Do you remember? Do you remember this one? Biochemical. What were some of the things we looked at there for evidence for evolution? Looking at DNA and looking at similar Proteins. What was another kind of odd thing we looked at that was still evidence for evolution, and it was biochemical? We all use cellular respiration. We all use ATP. We make certain proteins that are conserved. I'm looking for something else. Yeah, we all do that. Those are conserved processes. Conserved means it's maintained through several different species. We see similar genetic junk in some. Remember about the introns and the exons?
Oh.
Yeah, remember how it's like that trait's not even expressed, but we have that same DNA. Why is that? Probably because we had a common ancestor. You know? So there are things like that. Okay, good. What was this right here about? Exactly. I don't know why that one doesn't work. It's like all the other boxes short. What was some biogeographical evidence for evolution? Okay, location of certain species. Hello. So when there was a supercontinent, we see similarity, right, of species because they were joined. Because the idea would be the continents are apart. Was it because God created it here, here? Well, maybe. Ask him. Or is it possible, or her, or is it possible that they evolved in that one location where they were close together, and as it spread apart, now here they are? And then we looked at things like, really simple example to use all the time is mammals. And we know that mammals evolved about 65 million years ago, and that's when the continents were starting to separate. And that's why we have this huge diversity of mammals on each continent. But what might have come before a mammal? Well, let's start all the way in the ocean. So we have things that are aquatic, like fish. Then who would have evolved from the fish, maybe? Before your reptiles. Amphibians, and from the amphibian, reptiles, and from the reptiles, mammals. Yeah. So before mammals, you might see some plant species that were along. Remember when we look at the geologic time scale? Yeah. What era are we in right now? Cenozoic. And it's now back 65 million years ago. Remember how we drive that? Oh, remember how we remember that? Because we drive, right? Remember how we drive that? Okay. And then before the Cenozoic was the? No. Mesozoic. Mesozoic in the middle. Okay? So we have Mesozoic right here. Cenozoic, Mesozoic. So Mesozoic ended 65 million years ago, right? Because that's when the Cenozoic started. And then we go back to what? Well, Paleozoic's before that, but how far back does the Mesozoic go? 245. Because we remember in school, yeah, school gets out. And then before that was Old and Paleozoic. When did that start? 570, maybe? And then before that was Precambrian. And that's because the first period in Old and Paleozoic is the Cambrian period. So let me just kind of bring you through that, because I know you'll remember. Okay? Remember Precambrian time? When's the Earth start?
4.69.
Yeah, 4.5, somewhere in there, right? How many years ago? Billions, right? Okay. How? It's okay. All right, so 4.5 billion years ago. So at first, Earth, crust, rocks, right? First atmosphere lost. Second atmosphere we actually kept, but it was what kind of environment? Reducing environment. Remember that? There was no free oxygen, which was conducive to building. All right, and so then remember we said what came first? Remember we had where would the monomers have come from? Remember that? Oh yes. And remember outgassing from volcanoes, lightning, radiation, molten magma. Remember that? To give us our what? Monomers, right? And remember there was an experiment done? Yes. Miller. It's Miller time. And he was supporting Operan's hypothesis about that. And there was remember just within a week's time, he put the early gases in an apparatus and spun them around with electrical shocks and heated water, and then he pulled out the building blocks, right? Amino acids and simple sugars, right? And then you would need to hook those together to go from monomers to what? Polymers, and possibly the energy at the time, or clay, which can hold energy and act like a catalyst, could have done that, right? And we talked about protenoid microspheres and how hot proteins, when they cool, remember how they can form those spheres, and Operant's quasarbit droplets. You're looking like, no. Does it sound familiar at all?
Yeah,
It was a holiday.
Yeah, Haldane, good. All right, and Karen Smith, remember with the clay? Remember there was a debate, what came first, the protein, right? Who acts as the enzymes? Or, yeah, Fox, protein. Don't come together. All right, so we looked at, did the nucleic acid come first, who codes for it, or was it the protein? And what do we really think happened? Well, RNA. I'm hearing it. RNA first hypothesis. Good. Because we know that RNA can store a genetic code, but ribozymes can act like catalysts. So it could have been like an enzyme and store a code. Yeah. Yeah. There you go. I got a word. I'm throwing a word out there and seeing if it's going to stick. All right. Swedish. All right. So we were looking at evolution. We were saying, okay, so we got to get the polymers, and we got to get our first cell, right? We need a barrier. Remember, it could have been like a phospholipid, could have been a protein, or a mix of both. And our first cell, it needed to be self-reproducing, really. It needed to make more cells. And our first cells probably were what? They were anaerobic, right? Because there was no free oxygen. And they were pro or eukaryotic? Prokaryotic. Good. Were our first cells archaea? No. Because it's wrong name, right? They named them poorly. They didn't know. Because they're really more closely related to eukaryotes than they are prokaryotes, right? So prokaryotic cells. And then we look at the protista. And we have fungi, like fungus-like unicellulars. We have plant-like and animal-like. Then those prokaryotic cells, eventually we had a certain bacteria. Oxygen revolution, who did it probably? Cyanobacteria. All of a sudden they're putting oxygen out in the air, and that oxygen then allowed for what kind of respiration? Aerobic respiration. It allowed for oxygen to create a what layer? Ozone layer to protect, so we could crawl out of the water and get on land. Multicellular organisms, you with me? Okay, and we're still not even to the Paleozoic era yet. Okay, so at the end of the Precambrian time, you have small, like, invertebrates and algae and things like that. Then when we get into the Paleozoic era, that's your big change in the Paleozoic era, because you're going to go from invertebrates all the way up to early reptiles, right? Remember that? And then you're going to go from plants all the way up to early gymnosperms, right? Okay, then the Mesozoic era, you're going to have predominance of reptiles, and then move into early mammals, and plants are going to move into early angiosperms, and then in the Cenozoic era, we have all of our flowering plants, we have mammals, right, the last 30 seconds of our 24-hour day. Is this sounding familiar? You might want to go back and look at that. Okay?
Yeah,
Maybe. So biogeographical, that was this right here, dating, geologic time scale. And so when the mammals, so we had that huge diversity about 65 million years ago when they evolved at the beginning of the Cenozoic era. Anatomical evidence. 3 pieces of evidence, anatomical. Homologous structures. We're going to ignore what kind of structures? Analogous. Because analogous are probably as a result of what kind of evolution? Convergent. Good. Right? So they're just handling their environment in the same way. Homologous structures have the same anatomy. They might use them for different functions, but they have the same anatomy, right? So they probably had a common ancestor. What was another one you were going to tell me? Vestigial structures. What would be an example of vestigial structure? Tailbone, those kind of things. Not your backbone. You probably don't want that. Subphylum vertebrata. Okay? And what was our third one? Embryological. Embryological structure. So you can see similarity development could show similarities. Also, you can almost see evolution on fast forward as you go through. Okay, sweet. And that here, fossil record, we talked about this. There's some transitional fossils. The horse, that's always a good example. We know biogeographical. Can I go through that? We hit that already. We talked about Pangea. Australian marsupials, we've done this, we've done this. Are you comfortable with that? I hit it already. We did vestigial structures. Why might this salamander not have any eyes? Yeah. He doesn't need them, right? But he has remnants of eyes, so that shows you where he could have come from. Good. And here's embryological development, here's biochemical, and here is a question. And by the way, my friends, perfect timing, because we got through one enduring understanding. One. Another 54 to go.
No.
But it's fun.
If you did one a day, right? We have March, April. Yeah, that would give you 60. Guys, look, think about it like that. If you did an enduring understanding away,
It would work.
it would work.
It would also be really reasonable. Like it just,
One enduring understanding a day.
Animals, because they're the same.
I'm trying to read it. Wait, I didn't. I don't know. No, wait.
Your name or mine.
I remember these clicker questions.
You do remember them?
Practice test. How with or like on the first those two.
Dog kids.
Can I end it? Are you close? Let's just end. Let's just end. Flipper of a dolphin and the fin of a tuna are analogous because they have the same environmental response. They're swimming through water, but they don't have the same anatomy. That's why they're not homologous, but 4 of you said so. Look at their screen. If they're not showing you their screen, hey,
I'm mixing my.
hey. All right, come back to me. Wing of a bat, arm of a human. What is that? Homologous. They have the same anatomy. Look right here. Wing of a bat, arm of a human, humerus, radius, ulna, carpals, metacarpals, phalanges, same bones. It's just phalanges, right? Okay. All right. Come back. Oh, don't restart. Okay, good. All right. 4 of you struggled with that. Look at their screen. Counsel them or shame them. I don't care. One or the other. Again, 4. Are these the same 4 people?
That was me. I missed. I didn't get
It could be. I wouldn't know.
So, why is a dog kidney not vestigial?
Still need it.
Yeah, still need it.
Nephrons.
What? Natural selection occurs when the better adapted members. You all got that. Good job. All right. So our next review, we will pick up organisms are linked by lines of descent from common ancestry. That sounds like fun. Okay, we'll pick up there next time. Good job, you guys. Super proud of you. 54 more to go. 54 more to go.